So let's see regarding prokaryotic cell. In the prokaryotic cell, it is represented as bacteria, blue-green algae, mycoplasma. All the three will come under the prokaryotic. So coming to the mycoplasma, the size is 0.3 micrometers and bacteria is 3 to 5 micrometers. They are generally smaller and multiply very fast. Multiply rapidly. So and than the eukaryotic cell, and they very greatly range in size and shape. The four basic shapes of the bacteria are, so let me show you. If it is rod shaped, so let me start from here. If it is round shaped or spherical, this is cocci, and individually we call it as coccus. This is diplococcus, and this is streptococcus. So strepto means chain. This is staphylococcus. And coming to the bacillus, which is rod shaped, bacillus, and plural is bacilli, and we have diplobacilli, and here we have streptobacilli. Okay, and we have Vibrio. Example for Vibrio is Vibrio cholera. So this is comma shaped bacteria, and we have spirillum, spiral shaped bacteria. So the organization of prokaryotic cell is fundamentally similar. So even though the prokaryote will exhibit wide variety of shapes and sizes, organization will be similar. All prokaryotic cell will have cell wall except mycoplasma. So except mycoplasma. And coming to mycoplasma, usually mycoplasma have two things we have to remember. One is it is lack of cell wall. Other thing is it is anaerobic. Right? And coming to the next thing. So the ultrastructure of bacterial cell will be seen with high magnification microscopes, like 1500, 1 lakh times, 50,000 times. The magnification has to be there, like electron microscope. So usually the ultrastructure of prokaryotic, the very first thing that is present is cell envelope. Coming to the cell envelope, bacterial cells have complex cell envelope. Cell envelope consists of tightly bounded 3 layers. So we'll start from outer, glycocalyx, cell wall, plasma membrane. Bacteria is classified into two groups based on the cell envelope. So how do they respond the staining procedure? So by gram staining. Mainly this experiment was conducted by Christian Gram. So those who take up the stain is gram positive, and it is two things. Those who take up the stain is gram positive. Those who do not take up the stain is gram negative. So coming to how the staining process will determine, so by physical and chemical properties of the cell wall, by checking or by detecting peptidoglycan. Remember one more very important thing, gram positive consists of teichoic acid in its cell wall. And the very first thing is, so by detecting the peptidoglycan which is present in the cell wall of gram positive. So in gram positive we have peptidoglycan. Even here also it present peptidoglycan. But very small layer of it is dissolved when alcohol is added. That is why cell loses the initial color of primary stain. So the gram positive bacteria retain the crystal violet and thus stain it the violet. So gram positive is violet color. While the gram negative bacteria do not. So do not have. By adding safranin, gives pink color. Pink color. So pink color is gram negative and gram positive is violet color. If you see, so first initially take a gram positive bacteria and take a gram negative bacteria. So first initially the fixation. After fixation we have to add crystal violet. Then this both will be attaining the crystal violet color. And again now it is being treated with iodine. Okay, again iodine will give the same crystal violet color. And next again decolorization. Decolorization. Here this will come into normal color. But this will not lose the color, this will be still violet. And counter stain with safranin. Right? So when we got counter stained with safranin, so this is violet color. and this is pink color. So this is gram positive. And coming to the next thing, this is negative. Okay, so let's see the other characteristic features. So coming to the next thing, glycocalyx. So which is made up of polysaccharides. Glycocalyx, which is made up of polysaccharides. This is gelatinous sheet which is present around the cell wall. The slime layer, if it is loose, we call it as slime layer. Loose, it is slime layer, which is prevents the bacteria from dehydration. Prevents from. Next, if it is rigid, then we call it as capsule. They protect bacteria from antibodies and phagocytosis. Mainly, they protect from antibodies. And next, cell wall. Cell wall, we already know it is made up of peptidoglycan, or you can also call it as murin or lipopolysaccharide. Actually, the peptidoglycan is made up of N-acetyl glucosamine and N-acetyl muramic acid. Naga and Nama remain like that. N-acetyl glucosamine, and N-acetyl muramic acid. So which forms the chains? The chains are interlocked by tetrapeptides. So in gram positive cell, 90% will be, so N-acetyl glucosamine, N-acetyl muramic acid. In gram positive, 90% is peptidoglycan. %10 is lipopolysaccharide. In gram negative, reverse, 10% is peptidoglycan. 90% is lipopolysaccharide. Next, plasma membrane. So it is the innermost to the cell wall. It is selectively permeable. So made up of phospholipids and proteins. So two things. So next thing is mesosomes, parts of cell, mesosomes. So mesosomes are infoldings of the cell membrane. It is characteristic feature of prokaryotes. So it is in the form of vesicle. Vesicle means like this, tubule or lamellae. This is vesicle, this is tubule, this is lamellae. So it will be helpful in cell division, DNA replication, and mainly for respiration, secretion. So mainly also to increase the surface area. Especially in autotrophic bacteria, these mesosomes we call it as chromatophores. Next thing we have flagella. So three things: flagella, pilli, and next fimbriae. Coming to first thing, flagella. Flagella is motile or non-motile? If motile, there is a thin filamentous extension from the cell wall called flagella. Flagella is made up of a protein called very simply flagellin. And it has three parts: basal body, hook, filament. The basal body is embedded in the cell envelope actually. The hook is the curved structure connect to the basal body with flagellum. So the filament is long tube which causes the turbulence in the liquid medium by rotatory movement. So bacteria shows wide range in number and arrangement. So if you see, this was bacterium, you will be having the basal body, hook and so we will be having the basal body, hook, filament. Okay. And next, pilli and fimbriae. Pilli, which is the surface area of bacteria which is not involved in motility. These are the two structures which is not involved in motility. So this is made up of a protein called pillin, and they are found in both gram positive and negative bacteria. And are antigenic, that allows cells to recognize other cells and bacteriophages. Sex pilli is usually found in gram negative bacteria, sex pilli, and forming a bridge for mainly between two bacteria, that is conjugation. The fimbriae is like small bristle like structures, bristle like fibers. So sprouting out of the cell surface, they helps in anchoring the cell, mainly anchoring to rocks, host tissues. Coming to the other materials, cytoplasm and its contents. So the cytoplasm contains the viscous, very viscous, because it contains carbohydrates, proteins, fats, lipids, enzymes, coenzymes, and membrane bounded organelles like mitochondria, Golgi apparatus. Lysosomes are actually absent. So it consists of ribosomes. The ribosomes are associated with plasma membrane as well as with mesosomes. They are like 50s and 30s is equal to 70s ribosomes. They are polyribosomes, or we otherwise call it as polysomes, with numerous ribosomes attached to the mRNA. From polysomes, number of copies of polypeptide is made actually. Okay, so next we have the nucleoid or incipient nucleus, we otherwise call it as incipient nucleus. Actually in this the DNA is a genetic material. So we have the bacterial, only one bacterial chromosome is there, that too double stranded single circular DNA associated with positively charged non-histone proteins. And RNAs. And next thing is we have plasmids. This is also small circular DNA. Small circular double stranded DNA. Apart from nucleoid DNA. So the smaller DNAs we call it as plasmids. The plasmids replicate independently of the main DNA. So the plasmids may carry genes for specific functions, like genes for antibiotic resistance, and N2 fixation genes. Next, finally we have inclusion bodies. Coming to the inclusion bodies, so reserve materials are actually stored in the cytoplasm in form of inclusion bodies. They are no membrane bounded organelle. They flow freely in the cytoplasm, that is phosphate granules, cyanophysian granules, and glycogen granules. And gas vacuoles are found in green and purple green photosynthetic bacteria. Gas vacuoles. This helps for buoyancy. Green and purple green photosynthetic bacteria. Right. Let's see the process of gram staining. So coming to the gram staining process, so the ingredients we have to take is slide, Bunsen burner, bacterial culture, crystal violet, iodine, ethanol, water, saffronin. So initially we have to take a slide and we have to fix that slide. So we have to fix that slide with the bacterial culture by taking a drop of bacterial culture on the slide. And now we have to heat that Bunsen burner with the help of Bunsen burner, so the bacteria will fix on the slide. Later we have to add the crystal violet. After adding the crystal violet, all the bacteria will be taking the stain. So afterwards what we have to do is we have to wash with water. Some of the stain will be lost. And later we have to add the ethanol, iodine to fix the stain. And laterwards we have to wash with ethanol again. So the gram negative bacteria will be losing the stain, and later on we have to add the saffron in. So that's how the stain will be fixed, and again we have to wash with water. And usually if it is gram positive, it will lose the saffron in stain and it will show the gram positive. If it is gram negative, it will not have any blue stain, only red stain will be there. So this is gram staining procedure.